Lac operin. The lac operin, a key model for prokaryotic gene regulation, intricately orchestrates metabolic adaptation by allowing bacteria to efficiently utilize lactose for energy production when glucose is limited. Beyond negative regulation by the repressor, the operon undergoes positive regulation through the catabolate activator protein, CAP, fine-tuning gene expression based on glucose and cyclic AMP levels. Its structural features suggest an evolutionary adaptation, providing bacteria with a competitive edge in diverse environments. The lac operin's significance extends to biotechnology, where the LAX gene serves as a reporter for gene expression, facilitating research and experimentation. The operin's principles, while foundational in prokaryotes, are evolutionarily conserved, contributing to the understanding of gene regulation across biological systems. Overall, the lac operon holds profound implications in metabolic regulation, gene expression dynamics, clinical applications, and technological advancements. Regulation of gene expression. Both prokaryotic and eukaryotic cells tightly regulate gene expression to ensure proper functioning. The lac operin, a model in prokaryotes, exemplifies this regulation by controlling the synthesis of specific proteins in response to environmental cues. Lac operin as a model. The lac operin, a paradigm of prokaryotic gene regulation, provides valuable insights into the mechanisms by which organisms tightly control the synthesis of specific proteins. In prokaryotic cells, such as those found in bacteria, the lac operin serves as a model system for understanding how genes involved in lactose metabolism are regulated. Lac operin components. Comprising several key elements, the lac operin includes structural genes, LAX, LACI, LACA, a promoter, an operator, and a regulator. The structural genes encode enzymes necessary for the breakdown and utilization of lactose. The promoter is the region where RNA polymerase binds to initiate transcription, and the operator acts as a switch that can turn the operin on or off. The regulator gene produces a repressor protein. Proteins in lac operon regulation. Central to the functioning of the lac operon is the interplay of proteins. The repressor protein, synthesized by the regulator gene, acts as a molecular switch. Under normal conditions, the repressor binds to the operator, impeding RNA polymerase from transcribing the structural genes. However, in the presence of lactose, an inducer binds to the repressor, altering its configuration and preventing it from blocking the operator. This allows RNA polymerase to initiate transcription. Mutants and lac operin function. Mutations within the lac operon can have profound effects on its regulatory dynamics. These mutations may occur in the structural genes, promoter, operator, or regulator. By studying these mutants, researchers gain insights into the fine-tuned mechanisms that govern gene expression. Mutations may disrupt the normal functioning of the lac operin, leading to aberrant responses to lactose concentration changes. Significance of lac operin. Beyond its theoretical significance, the lac operin holds practical importance. It illustrates how bacteria optimize resource utilization by selectively expressing genes involved in lactose metabolism only when needed. This dynamic regulation allows bacteria to adapt efficiently to their environment, reflecting an evolved strategy for energy conservation.
Types of DNA mutations. Embarking on the exploration of DNA mutations is akin to stepping into a captivating journey through the molecular intricacies that govern genetic variation. DNA mutations, these subtle yet potent alterations in the nucleotide sequence, act as the engines of evolutionary change, intricately shaping the diversity of life. In this comprehensive examination, we are about to dive into the intricate world of inherited changes, base substitutions, transitions, transversions, and the cascade of effects these mutations have on proteins. Additionally, we will navigate the external influences of mutagens and the cellular guardianship provided by DNA repair mechanisms, all of which contribute to the nuanced landscape of genetic diversity. Inherited change in nucleotide sequence. The foundation of genetic diversity lies in the heritable changes woven into the DNA fabric. These changes, passed down through generations, introduce genetic variations that underscore the richness of biological adaptation. Imagine our genetic material as an ancient tome, with each heritable change being a chapter, contributing to the evolving story of life. Base substitutions. Now, let's zoom in on the heart of genetic mutations, the intricate dance of base substitutions. This category unfolds into transitions, where nucleotides of the same category smoothly swap places, and transversions, a more profound exchange between purines, adenine and guanine, and pyrimidines, cytosine and thymine. Visualize this as a dance floor where the dancers, representing nucleotides, elegantly change partners, adding complexity to the genetic choreography. Transitions and transversions. The terms transitions and transversions refer to specific types of base substitutions, which are changes in the nucleotide sequence of DNA. These substitutions play a pivotal role in genetic variation and evolution. Transitions. Definition: transitions are a type of base substitution where a purine nucleotide, adenine or guanine, is replaced by another purine, or a pyrimidine nucleotide, cytosine or thymine, is replaced by another pyrimidine. Example: if an adenine is replaced by a guanine, a G, or a cytosine is replaced by a thymine, CT, these are considered transitions. Characteristics: transitions involve the exchange of nucleotides within the same chemical category, purine for purine or pyrimidine for pyrimidine, and they are often considered more conservative changes in the DNA sequence. Transversions. Definition: transversions are a different type of base substitution where a purine is replaced by a pyrimidine or vice versa. Example: if an adenine is replaced by a cytosine, FC, a guanine is replaced by a thymine, GT, or any other combination involving the switch between purines and pyrimidines, these are considered transversions. Characteristics: transversions result in a more substantial change in the DNA sequence. They involve the replacement of nucleotides from different chemical categories, introducing greater diversity compared to transitions. Significance. Understanding transitions and transversions is crucial in the study of genetic mutations and their impact on evolutionary processes. The distinction between these two types of substitutions provides insights into the mechanisms that drive genetic diversity and the potential functional consequences of such changes. Transitions and transversions are also relevant in the context of molecular evolution and phylogenetics. Analyzing the ratio of transitions to transversions, known as the transition/transversion ratio, can be informative in assessing the selective pressures acting on a particular gene or region of the genome. Additionally, this ratio is used in constructing phylogenetic trees and understanding the evolutionary relationships among different species. In summary, transitions and transversions are key components of genetic variation, and their nuanced differences contribute to the intricate tapestry of evolutionary processes within the genome. Effects of base substitutions. These symphonic movements resonate through the protein coding regions, influencing the melody of genetic expression. Silent mutations, residing in non-coding regions, are like the unheard background notes, casting no impact on the orchestration of protein synthesis. Missense mutations, on the other hand, are akin to subtle alterations in the melody, a tweak to a single amino acid that may steer the function of the resulting protein. Meanwhile, the dramatic entrances of nonsense mutations serve as abrupt stops in the musical composition, leaving the narrative unfinished with premature stop codons. Reversions. In the intricate landscape of genetic mutations, the phenomenon of reversions unfolds as a compelling subplot. Reversions occur when a previously mutated gene, altered from its original sequence, undergoes a process of restoration, returning to its ancestral state. This genetic reset is not a common occurrence and typically involves the acquisition of additional mutations that effectively correct the alterations introduced by the initial mutation. From a scientific perspective, reversions are intriguing because they shed light on the plasticity and adaptability of the genetic code. The restoration of the original sequence implies a certain level of flexibility in the genetic material. The reversion process often involves a series of nucleotide changes, either substitutions or insertions, which collectively contribute to the restoration of the functional gene. Researchers have studied reversions as a means to understand the underlying mechanisms governing DNA repair and fidelity. The occurrence of reversions provides insights into the robustness of cellular repair systems and the ways in which cells can mitigate the impacts of mutations. Furthermore, the study of reversions contributes to our understanding of the mutational landscape and the potential evolutionary trajectories of genes over time. It's important to note that while reversions can be considered a form of genetic correction, they are not always advantageous. The restoration of the original sequence may or may not confer a selective advantage, depending on the environmental context and the specific function of the gene in question. In essence, reversions present a dynamic aspect of genetic mutations, showcasing the intricate interplay between mutational events, DNA repair mechanisms, and the adaptive potential encoded within the genetic material. This phenomenon adds depth to our understanding of the genetic narrative, illustrating the ways in which genes can evolve and adapt over time through the intricate ballet of mutational processes. Frameshift mutations. Frameshift mutations represent a significant and often dramatic alteration in the genetic code, where the insertion or deletion of nucleotides disrupts the reading frame during translation. Unlike other types of mutations that involve the substitution of individual nucleotides, frameshift mutations create a shift in the grouping of nucleotides into codons, the basic units of genetic information. Mechanism. The term frameshift is aptly named, as it describes the shifting of the reading frame, or the triplet code used by the cellular machinery during protein synthesis. In a normal, unmutated gene, the sequence of nucleotides is read in groups of 3, each triplet coding for a specific amino acid. When nucleotides are added or deleted, the entire reading frame is thrown out of sync. This creates a ripple effect, altering the grouping of nucleotides and, consequently, the sequence of amino acids that make up the protein. Consequences. The consequences of frameshift mutations are profound. Since the genetic code is read in triplets, the insertion or deletion of even a single nucleotide shifts the reading frame, leading to changes in every subsequent codon. This results in a completely different sequence of amino acids downstream from the mutation site. Protein structure and function. The altered amino acid sequence often results in the production of a non-functional or malfunctioning protein. Proteins are intricate molecular machines, and their structure is intimately tied to their function. Frameshift mutations can introduce premature stop codons, leading to truncated and usually non-functional proteins. Even if a full-length protein is produced, the altered amino acid sequence can disrupt the three-dimensional structure essential for proper function. Occurrence and impact. Frameshift mutations can occur spontaneously due to errors during DNA replication or as a result of exposure to mutagenic substances. Their impact on the organism depends on the specific gene affected and the role of the protein it codes for. In some cases, frameshift mutations can be lethal, while in others, they may result in genetic disorders or contribute to the development of certain cancers. In conclusion, frameshift mutations are pivotal events in the genetic narrative, altering the course of protein synthesis and often resulting in significant biological consequences. Their understanding provides insights into the complexity of the genetic code and the delicate balance required for the proper functioning of cellular processes.
Mutagens. As we navigate the genetic narrative, external forces enter the scene. Mutagens, chemical substances, radiation, and certain viruses become influential characters in our genetic plot, adding an element of unpredictability. They influence both the rate and character of mutations, contributing to the dynamic nature of genetic variation. Mutagens, in the realm of genetics, are external agents that have the potential to induce mutations in the DNA sequence. These agents can introduce changes ranging from single nucleotide substitutions to large-scale alterations, impacting the genetic blueprint of an organism. Understanding mutagens is essential for unraveling the complexities of genetic diversity, evolution, and the factors that contribute to the dynamic nature of living organisms. Types of mutagens. Chemical mutagens. Definition: chemical mutagens are substances that, upon exposure, can interact with DNA, causing alterations in its structure. Examples: aromatic amines, alkylating agents, and certain industrial chemicals fall into this category. Mechanism: chemical mutagens may modify individual nucleotides. form covalent bonds with DNA strands, or induce chemical reactions that result in base substitutions or other types of DNA damage. Radiation mutagens. Definition: radiation mutagens are agents that induce mutations through exposure to various forms of radiation, including UV light and ionizing radiation. Examples: ultraviolet, UV, light, X-rays, and gamma rays are common sources of radiation mutagenesis. Mechanism: radiation can cause DNA damage by generating free radicals or directly affecting the structure of DNA molecules, leading to mutations during repair processes. Biological mutagens. Definition: biological mutagens involve living organisms or their products that induce mutations. Examples: certain viruses and transposable elements, also known as jumping genes, can act as biological mutagens. Mechanism: viruses can integrate into the host genome, leading to mutations during the integration process. Transposable elements can move within the genome, causing genetic rearrangements. Impact on genetic variation. Genetic diversity. Mutagens contribute significantly to genetic diversity within populations. The introduction of mutations is a driving force behind the evolution of species, providing the raw material for natural selection to act upon. Adaptation. Mutations induced by mutagens can confer adaptive advantages or disadvantages depending on the environmental context. Certain mutations may enhance an organism's ability to survive and reproduce in specific conditions. Cancer development. Exposure to mutagens is a known risk factor for the development of cancer. Mutations that disrupt the normal regulation of cell growth and division can lead to uncontrolled cell proliferation, a hallmark of cancer. Mitigation and repair. Cells have evolved intricate mechanisms to mitigate the effects of mutagens and repair damaged DNA. DNA repair pathways, such as base excision repair, nucleotide excision repair, and mismatch repair, play crucial roles in maintaining the integrity of the genetic code. These mechanisms act as guardians, continuously surveilling the genome and correcting errors introduced by mutagenic agents. In conclusion, the influence of mutagens on the genetic destiny of living organisms is profound. From shaping the diversity of species to contributing to disease development, mutagens are integral players in the ongoing saga of genetic evolution. The delicate balance between mutation, repair, and environmental influences highlights the intricate dance of life at the molecular level. DNA repair. Amidst this symphony of genetic variation, cells deploy an awe-inspiring arsenal of DNA repair mechanisms: base excision repair, nucleotide excision repair, mismatch repair. These intricate processes act as the unsung heroes, sentinels tirelessly patrolling the genome. They mend and maintain its structural fidelity, ensuring that the genetic narrative stands the test of time, much like a vigilant guardian preserving the integrity of an ancient manuscript. DNA repair mechanisms are the cellular safeguards that ensure the stability and integrity of the genetic code, constantly engaged in a meticulous dance. These repair systems play a pivotal role in maintaining genomic fidelity by identifying and correcting errors, damage, or mutations that occur within the DNA molecule. The intricate nature of DNA repair reflects the evolutionary imperative to preserve the accuracy of genetic information, vital for the proper functioning of living organisms. Types of DNA repair mechanisms. Base excision repair, BR. Function: BR primarily addresses small, non-helix distorting lesions, such as damaged or mismatched bases. Process: specialized enzymes recognize and remove the damaged base, and the gap is filled by DNA polymerase and sealed by ligase. Nucleotide excision repair, NER. Function: NER is involved in the removal and repair of larger lesions that distort the DNA helix, such as those induced by UV radiation. Process: enzymes recognize the distortion, excise the damaged segment, and the gap is repaired by DNA polymerase and sealed by ligase. Mismatch repair, MMR. Function: MMR corrects errors that occur during DNA replication, ensuring that the newly synthesized strand matches the template strand. Process: the mismatch is recognized, and the segment containing the error is excised and replaced with the correct sequence by DNA polymerase. Double-strand break repair, DSBR. Function: DSBR addresses breaks in both strands of the DNA double helix, which can be caused by various factors, including ionizing radiation. Process: there are two main pathways for DSBR, homologous recombination, which uses an undamaged sister chromatid as a template, and non-homologous end joining, which directly rejoins broken ends. Importance of DNA repair. Genomic stability. DNA repair mechanisms are crucial for preventing the accumulation of mutations that could otherwise lead to genetic instability. Maintaining genomic stability is fundamental for proper cellular function and the prevention of diseases such as cancer. Cell viability. Unrepaired DNA damage can result in cell death or malfunction. DNA repair ensures the survival and functionality of cells by promptly addressing issues that could compromise genomic integrity. Prevention of mutations. DNA repair mechanisms are the frontline defense against mutations caused by environmental factors, mutagens, and errors during DNA replication. By promptly repairing damage, these mechanisms mitigate the risk of permanent alterations to the genetic code. Adaptation and evolution. DNA repair also plays a role in the evolutionary process. Repair mechanisms can tolerate a certain level of genetic variation, allowing for adaptation to changing environmental conditions while still maintaining the overall stability of the genome. Challenges and implications. Inherent imperfections. While DNA repair mechanisms are highly efficient, they are not infallible. Some errors may escape repair, leading to the persistence of mutations. Cancer and DNA repair. Dysregulation or impairment of DNA repair mechanisms is associated with an increased risk of cancer. Deficiencies in repair pathways can allow the accumulation of mutations, contributing to the initiation and progression of cancer. Therapeutic applications. Understanding DNA repair mechanisms has implications for medical treatments. Some cancer therapies, for instance, exploit the vulnerabilities of cancer cells with impaired repair mechanisms. In essence, DNA repair mechanisms serve as the vigilant guardians of the genetic code, continuously surveying and correcting errors to maintain the intricate balance between stability and adaptability within the ever-evolving dance of life. Chemical mutagens, fascinating agents capable of inducing genetic mutations in living organisms, play a pivotal role in genetic and environmental studies. These substances encompass a spectrum of types, each with distinct mechanisms that impact the DNA structure. For instance, base analogues, such as 5-bromouracil, mimic DNA bases and may introduce substitutions during replication, thereby altering the genetic code. Alkylating agents, exemplified by ethyl methane sulfonate, EMS, add alkyl groups, inducing point mutations or strand breaks. Deaminating agents, like nitrous acid, remove amino groups, prompting base pair changes, and intercalating agents, such as ethidium bromide, inserted between base pairs, can cause DNA strand slippage during replication. The mechanisms through which chemical mutagens exert their effects are multifaceted. Direct damage involves the modification of DNA structure, disrupting the replication and repair processes. Indirect damage, on the other hand, results from the generation of reactive intermediates, such as free radicals, which can be highly mutagenic. As an example, polycyclic aromatic hydrocarbons, PAHs, in cigarette smoke generate reactive metabolites, contributing to the mutagenic effects associated with smoking. The consequences of these mutations vary, ranging from silent mutations with no apparent phenotype change to missense mutations. An illustrative example is the mutation in the TP53 gene, often associated with cancer. Nonsense mutations, such as those caused by the chemical 5-azacitidine, introduce premature stop codons, leading to truncated and often non-functional proteins. Frameshift mutations, exemplified by the insertion of a base analog like 2-aminopurine, arise from insertions or deletions, disrupting the gene's reading frame. Several factors influence the mutagenicity of these chemicals, including concentration, exposure time, and metabolic activation within the organism. The efficiency of DNA repair mechanisms, such as those involving enzymes like DNA glycosylases, also plays a crucial role in mitigating mutagenic damage. Understanding the implications of chemical mutagens extends beyond the laboratory. In cancer research, these agents are integral, as many carcinogens induce mutations. Examples include the association between exposure to aflatoxins, produced by certain fungi, and mutations leading to liver cancer. Moreover, the environmental impact of chemical mutagens raises concerns for ecosystems and human health, emphasizing the need for comprehensive studies. In the realm of drug development, a thorough grasp of mutagenic effects is essential for evaluating the safety of pharmaceuticals. As an example, the chemotherapy drug cyclophosphamide is known to be metabolically activated to form mutagenic intermediates. This interconnected web of knowledge surrounding chemical mutagens underscores their significance in scientific disciplines, from genetics and toxicology to environmental science. Delving into their complexities reveals not only their potential dangers but also opens avenues for leveraging this understanding in diverse fields, contributing to advancements in medicine, environmental management, and beyond.